Go. All right, welcome everybody. Thank you for coming. First of all, I just want to say thank you guys for everything you do. We just had a debrief on a difficult patient. I know that we had in the corner there, that trauma patient. And for those of you that couldn't make it, I think it was really productive. We were able to talk through some things, but it really drove home to me, from the bottom on up, what you all do and how important that is for the delivery of care. And that goes from facilities management, corpsmen, nurses, everyone. We just come up with the plan. But as I was telling Lieutenant Makis yesterday, I was like, if it was up to me to actually deliver the care, like everyone would die. Because you guys are the people that put the rubber on the road and make things happen. So I appreciate what you do. All that to say, we can talk about lactate. So this is kind of complicated, like biochemistry, a little bit mind numbing, but we got to kind of talk about that. And I look at this as like a scaffold or a Christmas tree, if you will, with which to hang the ornaments of knowledge on. We get Christmas theme, okay? All right, takers. So we're going to kind of go through this lactate production, how it gets produced, and it really has to do with like cellular metabolism in general. So I'll go through some basically just identifying some structures, but this is each cell has these components, but this is the cell membrane. And we know from our inotropes and our vasopressor talk that the cell membranes of certain types of cells have beta-2 receptors, correct? Can someone name me an inotrope or a vasopressor that hits beta-2? Epi, perfect, okay. So we've got a beta-2 receptor that stimulates something within the cell called cyclic AMP. You don't need to remember that. But basically what that stimulates is this glycogen store to get broken down to glucose. So we all need energy, correct, for cellular function to happen. So glycogen is stored in the liver and in the muscles, a bit in the kidneys as well, I believe, but mostly liver and muscles. So glycogen gets broken down into glucose. And we all know what glucose is. We check finger stick glucoses all the time. But we need glucose, right, for cellular metabolism and to create energy. So glucose then gets shunted into the mitochondria. This is a little mitochondria green. And has anybody heard the mitochondria being basically like the engine of the cell? So it's our motor. Basically, it is what is going to help us produce through this little TCA cycle ATP and energy for cellular functions. So typically what will happen is pyruvate will then enter the TCA cycle with the help of being broken down by this enzyme called pyruvate dehydrogenase, PDH, pyruvate dehydrogenase, which needs thiamine. Then it enters the TCA cycle, and it helps us form ATP and energy. I have outlined oxygen here because this is a very necessary step to basically offload an electron and allow this cycle to go around, okay? So this is an electron acceptor, and we make water basically, but you need oxygen for that to happen and to be able to create cellular energy. Okay, so where does lactate come into this? We'll start here, and this is basically aerobic lactate production because we're not talking about oxygen quite yet. But what are some things that stimulate beta-2? Somebody mentioned one of them. The beta-2 receptor. I heard epinephrine. Okay, what's our body's epinephrine? Anybody? It's called adrenaline, right? Adrenaline. And then we basically, so epinephrine is the form we make. And then is there another, like inhaled albuterol is a beta-2 receptor, is a beta-2 agonist, correct? So like albuterol. Okay, so all those things are going to stimulate beta-2, and they're going to kick off this cascade, okay? When you get significant, overwhelming stimulation of beta 2, say like continuous nevs. I've seen this happen before. Or really bad sepsis, where our catecholamines within our body, adrenaline, are stimulating beta 2. That's going to drive this way, okay? And basically, this is all in sort of a balance. And when you get too much of this, it starts to get shunted to lactate, okay, and lactate production. So we think about lactate as being something bad, and it is. This graph communicates the higher your lactate goes, the more likely you are to die, and that's pretty consistent. But lactate also should be thought of as almost an adaptive mechanism by the body under times of stress. It's almost what we view it in the ICU is essentially like a check engine light, right? The body's under stress for some reason. Why is that lactate elevated? But lactate can also be used as an energy source, and multiple organs use it adaptively. So the heart, typically at rest, only like 10% or so, but in times of stress can use up to 70% of its energy through lactate, and uses lactate for energy, and then through lactate dehydrogenase, shunts it back to pyruvate, and then it can kind of enter this cycle again. So the heart actually uses it constructively, at least initially. Lactate can also be used by the brain, usually only about 7 to 10% initially, but up to 25% in times of stress. So the brain can use lactate as an energy source, and again, can use LDH to take it back to pyruvate, and we can enter the cycle here. So that's like an adaptive good thing that lactate can do. The lungs under stress, especially in sepsis, can produce lactate, and can also use lactate. The kidneys and the liver are our big processors of lactate, or metabolizers of lactate. So they metabolize lactate. This is something called the Cori cycle. But basically, they take lactate, the liver and the kidneys, about 30% is cleared in the kidneys, 50% in the liver. But they can take lactate and through the Cori cycle produce glucose again, and then we can go in this direction again and produce energy. So lactate's actually, again, I'm not saying it's a check engine light. We want to make it go away, but at least in some instances, it's adaptive. Okay. So I say all these things because now we're going to have to figure out, okay, we've got one part of how is lactate produced, but what's another part where if I can't go around and around here and I'm running into an issue here, then I'm going to produce lactate here, okay? So the other big piece here is oxygen, correct? We have the equation of critical care, correct? Does anybody remember the equation of essentially critical care medicine, the delivery of oxygen equation? He's like scrolling back through his. It's like, yeah, I know it.
Equation. I titled it "Equation of Life." Delivery of oxygen equals cardiac output times heart rate.
Cardiac output is
heart rate times stroke volume.
Yep, heart rate times stroke volume times
percent
sat. Hemoglobin
plus PaO2
plus PaO2 times like 0.0003 or whatever. Yeah. Okay. So we've got the aerobic causes that we can shunt here and cause lactate production. Now we can basically have a delivery of oxygen issue problem that can cause lactate production. So are there some patients that have had a heart rate and stroke volume issue that we've seen in here that we've been trending lactates on? Like a pump issue? I'm thinking of a guy like here, who we had on dobutamine, cardiogenic shock. We were messing around with the lactates. We were trending those. We were trending SCVO2s, doing a lot of Guam type things. But he had a lactate elevation. His was a delivery issue, right? he could not deliver the oxygen. He could not mount a cardiac output to meet the demands of his tissues, right? So if he couldn't meet this for the tissues, they weren't able to offload that TCA cycle, right, and keep it moving. And what built up? This, and then it gets shunted to lactate. Make sense? Okay. There are some other things. Do you remember this column? Like, so hemoglobin, typically you've got to get severely anemic. So severe anemia, like I'm talking like 2, and abruptly.
Until then you compensate?
Depending on their other comorbidities and how good their heart is and pump and everything. But so low hemoglobin, it's got to be severe typically. But were there some other things we talked about that affect our percent SAT hemoglobin? Remember me talking about like cyanide poisoning, methemoglobinemia. So basically, if like your oxygen holds on to hemoglobin too hard and never gives off to the tissues, that could cause a delivery issue of oxygen. But then we talked about like the main ones here, right? And the main ones here were basically like decreased FiO2 if you're like on Everest or whatever. Never really happens here. Increase ventilation to perfusion, so like PE. Decreased ventilation to perfusion. That's like pneumonia or shunt. Somebody else help me out here. What are the other ones?
Hypoventilation.
Yep. And then we had talked about, decreased diffusion. So like a thickened membrane where you can't get oxygen across. Okay. So basically, if you remember this equation and what can affect it, then you can remember, all right, are we not delivering enough oxygen to the tissues? We're not allowing this cycle to offload the electron to make water. And then if this gets clogged up, then you're going to shunt and you're going to make lactate. Okay. So we got to work on increasing the delivery of this through manipulating these mechanisms, essentially. So there's some other things that are related to oxygen. Like what else could increase a lactate? Basically, due to a reduction in oxygen delivery to the tissues. What if you like clot something? So like I'll put here also like ischemic gut or tissue, just ischemic tissue. You can also transiently basically outstrip the ability for this equation to provide oxygen to the tissues in a normal fashion. Like, what if anybody heard of like lactic acidosis or lactate production? Like if someone were to sprint upstairs, and you would draw their blood after that and, really do like a max VO2 output, like up and down stairs, up and down stairs, up and down stairs till you get basically like lactate production, you're going to basically transiently outstrip your body's ability to provide the oxygen your muscles need for a short amount of time and produce a lactate elevation if you drew your blood at that time. And then your body's going to deal with it and clear it and you're going to be fine. So ischemic tissue, and so I'll say like seizure, exercise, things of that nature. Those are kind of the big ones in terms of oxygen delivery to this cycle, creating a backup where lactate gets produced. And then there's other like random things there's genetic things like inborn errors of metabolism within the mitochondria that do not allow you to do this function well. And so you can have like lactate production. Those are very rare. I don't want those to be in your mind in terms of like a major cause of like lactate elevation. I think what would probably be useful is if we now kind of talk about probably our most common thing is sepsis, right? And like, where does that kind of fit in? Because it's really kind of multiple ways. A lot of it is aerobic production through this mechanism, right? We've got a lot of catecholamines flowing when people are septic, maybe a little bit of that. So you've got some aerobic production, but also what sepsis on like a microvascular capillary level and cellular level does, typically like from bacteria exotoxins and things, is it kind of poisons the mitochondria. So, and it poisons this enzyme. It can make us deficient in this. So sepsis can really mess with like mitochondrial and cellular dysfunction and cause lactate elevation. It can do it through obviously the aerobic cause there. And then stick with something else. Those are the main. Those are really the main functions. So I mean, what do we try to do? We fluid resuscitate people. We give them antibiotics to reduce the bacteria, reduce the exo and endotoxins, and we resuscitate them. And hopefully, this dysfunction that we see at the mitochondrial level, also at the catecholamine level, and everything starts to calm down, and then our lactate starts to come down as well. But yeah, this is kind of how I think of lactate, and really more as, again, we know, as lactate starts to get over 4, the higher it climbs, really, 2's normal, but once we start to get over 4, the more we go up, the more our mortality goes up. So we've got to figure out where along this chain of events, aerobically or anaerobically, with a lack of oxygen, are we creating lactate? And what can we do to solve that issue? If it's ischemic tissue, we got to get that tissue out, or we got to reperfuse it. Reperfuse the heart, reperfuse the gut, take the gut out. This is usually a transient thing, like seizure or exercise. Once that is done, then people recover. But again, the delivery side of things, how do we solve this issue if it's an issue with hypoxemia and or reduction in delivery from the heart? And then that's typically will solve itself if we treat the underlying cause of like sepsis. What questions do people have?
How significant
Lactated ringers doesn't contribute to lactic acidosis. Dr. T might be able to answer this, but I was just reading the other night. It's basically like lactated ringers. It has to do with the acid-base status of it, and it's actually like the conjugate base of lactic acid. So it's like it's not a big contributor to, or a really contributor at all to what we think of as lactic acidosis. It's actually the go-to crystalloid for sepsis.
Actually, an excellent question. many physicians and many scientists raise that question. Why are we given lactated ringers when we have excess of lactic acid in the body? This is what I want you to think. Some of you go to the gym. Some of you work out too hard. You know that feeling when you wake up the next morning and your entire body is burning? Guess what? That's the lactate. Lactated ringers, however, doesn't contain lactate. In fact, using normal saline, which you probably noticed that I never use it in the United States, actually has more deleterious effect because of the renal hyperperfusion, you're going to generate more lactate. Plus, you're going to introduce more CL, and that would affect the NIMDF in the acid-base steps. So bottom line is lactated ringers does not increase the amount of lactate.
Yeah, a lot of people call it abnormal saline because it's really not a normal solution. It's actually acidotic, and lactated ringers is more pH neutral. So typically people already have an acidosis, so why are we giving them an acidotic solution in normal saline?
We'll still do it.
Yeah, I'd say caveat being typically the studies that show the deleterious effects is like more large volume resuscitation. And then I would say in people you're worried about sodium, like neuro patients, a lot of those people are going to be on, you know, some form of normal saline or hypertonic saline. And then you just know that, you know, you might give them a non-gap acidosis. But that's kind of how I think of like lactate in a nutshell. But it helps to understand how it's produced and how it's metabolized, mostly in kidneys and livers. So this is the last part I was going to make. Liver injury, acute liver failure, end-stage renal disease, kidney failure, that can make your clearance of lactate slower and impaired. So you kind of got to know the full picture and know how it's metabolized and produced to be able to understand like what's the problem I have to solve here to make my lactate go down and to clear it.
What was the percentage of clearance for liver and kidney?
Liver's like 50%, kidneys like 30%. Yeah, I've seen people with in like liver failure just not really clear their lactates. Other weird things that can cause lactate elevation are like leukemias, lymphomas, some like very large solid organ tumors. It's called like the Warburg effect, where the tumor cells essentially produce lactate. They're not like septic or anything; they just produce more lactate. But those are some like very fringe type things. I would say if you can connect lactate more to like what we see a lot of, which is basically like a deficit in the delivery of oxygen via one of these mechanisms here. We do see a lot of heart stuff, but we see a lot of this stuff too, hypoxemia, if that's severe, as well as the aerobic cause. And then knowing how does sepsis affect cellular metabolism. And it's really multifactorial.
Does that change the approach much? Or, you know, we have a lot of kidney, CKD patients and some good amount of liver patients. Does it change your approach on how you treat this for those?
I don't think it necessarily changes my approach. It just makes me like realize that
You keep that in mind.
You know, keeping it in mind. Just like I keep in mind, like, you know, like that one person just wasn't responding to dobutamine, and so I had to put him on epinephrine. He loved it, but understanding that I may not normalize my lactate, and it's okay if he's making urine, if his cap refill is good, if he's not cool and clammy anymore, if he's not showing me other signs of shock, then I'll accept, or I'll just stop watching the lactate. You know, if I've got all those other markers of end organ perfusion, that he's doing okay. So that's where it's, again, I know it's a little bit busy, but you need to understand the parts to say, okay, why, how is lactate produced, and why could my lactate be elevated, and what might I do to solve that reason for why it's elevated and mitigate that. Any questions? Good. Is everyone like, WTF's going on?